Hi, I'm Katie Butts-Dehm, your biology instructor this semester. Today we're going to look at scientific inquiry and the scientific method. So what is science? Science is the pursuit of knowledge and understanding of the natural and physical world. When you are trying to learn something about the world, something about the natural world, something about the physical world, when you're trying to understand it, then that is science. Maybe you're trying to understand how photosynthesis works. You're trying to understand how plants take carbon dioxide and sunlight and make glucose and other sugars and starches. Then that would be science. In science, you have questions or problems, and then you look for answers to those questions or solutions to the problems using the scientific method. This class is called Scientific Inquiry. Scientific inquiry is using evidence from observations and investigations to create logical explanations and to answer questions. So scientific inquiry is going to be a little bit different than the scientific method and a little bit different than science. So we're going to look at the differences between scientific inquiry and scientific method. That's a little bit harder. But science is just more general, a more general definition. So scientific inquiry includes all the diverse ways in which scientists study the natural world and propose explanations based on evidence and derived from their work. So these are the ways that scientists study and the ways that they learn about the natural and physical world. That is scientific inquiry. Scientific inquiry also refers to the activities through which students develop knowledge and understanding of scientific ideas, as well as understanding of how scientists study the natural world. So here is scientific inquiry, and you're looking at all of the different ways that you learn about science. So communication, question, observation, defining, focusing, investigating, developing. So all of these different ways that you can study about science, that is scientific inquiry. And here is a scientific inquiry circle. So it pretty much is the same thing. You reflect. So do my findings prove my hypothesis? Was I right? Have I changed my original hypothesis? Things like that. You test. So that basically is setting up your experiment. And then your observation, you're observing your experiment. And you're recording your results. So your question that you ask, your hypothesis, right here. So all of these different things are involved in scientific inquiry, and it's more of a cycle. Now, the scientific method is a process involving a series of steps used to answer a question or solve a problem. So the scientific method is this process. It is a step-by-step process. You ask a question. You state the hypothesis, which is your educated guess to the answer of the question or problem. You form and conduct an experiment, design and conduct an experiment. You analyze the results and you make a conclusion. So those are specific steps that you follow with the scientific method. So the steps of the scientific method, make sure that you know these steps, know what they are, know them in order. Number one, you have a question or a problem. So an example might be, which fertilizer works the best for tomato plants? Another example of question or problem you might have would be, does this newly developed headache medication actually relieve headaches? So there's your questions, and that's your first step of the scientific method. So for the second step, what you do is you research the problem. So you don't just make up a hypothesis without understanding anything about the problem. So you may look into information about tomato plants, their nutritional needs, how fast they normally grow, other needs besides nutritional needs, fertilizers, sunlight, things like that, amount of water. And then you also may research this other problem the causes of headaches and the current treatments that relieve headaches. The next thing you do is you formulate a hypothesis. A hypothesis is an educated guess about the answer to the question or the solution to the problem. So you're going to guess what the answer is. You're going to guess, does this fertilizer work good with the tomatoes? Maybe which fertilizer works best? Does this headache medication work better than just a placebo? But the thing about it, it's not just a guess, it's an educated guess. So you definitely needed to do your research beforehand. So here is our hypothesis for our first problem. Fertilizer B is expected to work the best because it's highest in calcium, magnesium, and potassium. Deficiencies of calcium, magnesium, and vitamin D are commonly found in tomatoes. So that is our first hypothesis. The hypothesis to the other problem would be the new headache medication is expected to relieve headaches because it contains the herb feverfew, which has anti-inflammatory effects and is commonly used as an herbal remedy for headaches. The next step of the scientific method is going to be to design and conduct an experiment to test the hypothesis. Now, it's really important to know a lot about designing the experiment. First of all, experiments should have a large sample size. If you try a headache medication on one person and it works on that one person, well, that one person may just feel better because they took something that might be a headache medication. Maybe they got a placebo, which is like a sugar pill. It doesn't matter. It might just be a psychological thing. Maybe the new headache medication only works on 3% of people. So you need a large sample size. You also need to have a control group. And a control group is a group that's identical to the experimental group except for the variable being tested. So with the tomatoes, you might have maybe 5 different fertilizers, or maybe just one, but let's say you have A, B, C, D, and E are all different fertilizers, and then you have a group of tomatoes that doesn't get any fertilizer, but it gets the same amount of water as all the other tomato plants, the same amount of sunlight, the same type of soil, same type of air and temperature. Everything is the same except for that variable that's being tested. For the headache medication, you generally will have a group of people that are taking a headache medication, and then you'll have a group of people that are not. The group of people that are not taking the headache medication, those are the control group. And you might just look at people in general, but then you may look at, for certain medications, people of a certain age, and really look at different things of it. But overall, you have a group of people that are testing the actual medication and a group of people that are having a placebo, which might be a sugar pill or a pill with nothing in it. A variable is any part of the experiment that can change or vary during the experiment. So before we were talking about the amount of sunlight, the amount of water, the type of soil, the temperature. All of those are variables. Now you need to make sure you keep straight the different kinds of variables. An experimental variable, that is the variable that's changed or the one that's tested. So in your tomato plants, all of the other variables can be controlled remain the same, but the only thing that changes is the type of fertilizer, or the control group doesn't get any fertilizer at all. Now you have the independent variable and the dependent variable. The independent variable is the same thing as the experimental variable. And look, they both start with vowels. But independent and experimental variable are the same. The independent variable is the variable that's being changed or tested. The dependent variable depends on the independent variable, okay? So, for example, this might be the height of the plant, okay? So maybe you give a different fertilizer and the plant grows taller. So the dependent variable, the height of the plant, changes because of the independent variable. So here are types of variables. Independent variable, the type of fertilizer that's given. There's fertilizer A, B, C, D, and that one just gets plain water. The other ones get water with the fertilizer. Your dependent the dependent variables would be the height of the plants, the number of leaves, the number of tomatoes. Maybe you might weigh the tomatoes and get the size of tomatoes. The controlled variables, now we haven't mentioned those, but these are the variables that all stay the same. This is the type of plant, so they're all going to be tomato plants, but not only just tomato plants. If maybe they're all going to be grape tomato plants. I like grape tomatoes, so they might all be grape tomato plants, or maybe they're all Roma tomato plants, or whatever type of tomato you like. But they're all the same type of tomato plant. They all get the same amount of water. They all get the same amount of sunlight, temperature, soil type, soil pH, and again, type of plant. And then you have the control group. That those are the only really variables there. So this is probably what I just said. Several types of fertilizer are being compared. They're being tested on tomato plants. The experimental variable is the type of fertilizer. All the other variables, the amount of water, the amount of sunlight, temperature, soil type, pH, all of those are kept the same, so those are control variables. And the control variables are different from the control group. The control group is a group of tomatoes that doesn't receive any fertilizer. All these other variables between this group and the others are all going to be kept the same. And here we didn't mention the dependent variable, but the dependent variable again is the height of the plants, maybe the number of leaves, the number of tomatoes, I would weigh the tomatoes. And here with the headache medication, the experimental variable is whether or not the person is given the headache medication. The control group is the group of people that are being given the fake medication, which is called a placebo, instead of the actual headache medication being tested. Usually you don't tell these people whether they're getting the headache medicine or the control, or the fake medicine, the placebo. Really, I don't think you almost ever tell them. So experiments should be controlled. And we talked about this already. In a controlled experiment, all the variables are the same except for the variables being tested. So your independent, light, color, dependent, plant height. In some experiment, you can actually give them a different color light. Like this one, you have yellow light versus blue light. Control variables, water, soil, temperature, and amount of day and night. And this is going to be the same for all the plants, and that means your experiment is controlled. And this is the one that the variable that you change, yellow or blue light, the independent or experimental variable, and then the dependent variable, the plant height. And look, they could have done number of leaves too. So here would be an example of a controlled experiment. 100 tomato plants are grown from seeds. So they're all planted in the same soil at the same pH. They're all given the same amount of water and sunlight. They're kept at the same temperature. They're grown for the same amount of time. Whether it's 1 week, 2 weeks, 5 months, they're all grown for that same amount of time. The plants that are given fertilizer are all given the same amount of fertilizer. So here we have 100 tomato plants. They're divided into 5 groups of 20 plants. So just one plant of each isn't going to be very good. You might just have a tomato plant that's a dud. So group 1's given Miracle Grow. Group 2's given Job's tomato fertilizer. Group 3 is given Epsoma tomato-tone organic fertilizer. Group 4 is treated with Neptune's Harvest vegetable fertilizer. Group 5 is not given any fertilizer at all. The heights of the plants, number of leaves, and number of tomatoes for each plant were recorded every week for 120 days. So that's what, about 4 months. The total number of tomatoes for each plant were also recorded. And again, if I had to change this, I would get the weight of the plant and the weight of the tomatoes separate. That is a controlled experiment. So experiments should be unbiased. By unbiased, this means that the opinions of the scientists conducting the experiments or of the human experimental subjects, they might unfairly influence or bias the experiment, and they should not, okay? The opinions of the scientists and the opinions of the experimental subjects should have no effect on the experiment at all. Otherwise, it's not really a valid experiment. So there's something called a single-blind experiment. This is an experiment in which the information that may influence the participants of the experiment is withheld from the participants until after the experiment is complete. Okay, so with the subjects in the headache medication study, they're not told whether they're receiving the experimental medication or the placebo. I was in one of these in college in a headache study, and I was paid $10 and an alarm clock to be in this study. So for like maybe a month, every time I had a headache, I would take whatever they gave me. Honestly, I never found out whether I had the headache pill or the placebo. The placebo is a substance or treatment which is designed to have no known therapeutic effect. People may feel better because they think they're given a medication, but and that's called the placebo effect. So you need to definitely compare a placebo, a group of people taking a placebo versus a group of people taking the medication, because overall you're going to get people thinking they're feeling better anyway because they had a placebo, but or 20% of them feeling better, or 50% or 70%. So that's what you need to compare the placebo group with the experimental group. So common placebos include sugar pills and saline injections. You know, honestly, I think that a sugar pill, even though back then that's what people used a lot, but a lot of people are affected by sugars. I know they're small, but maybe I'd want to put something in there that has even less effect than a sugar. So with the single blind experiment, then you don't have the experimental subject showing bias, other than maybe some placebo effect, but you might have the technician who's running the experiment might be biased. So a double-blind experiment is even better. So this is an experiment in which information that may influence the experiment is withheld from not only the research subjects, but also from the technicians performing the measurements until after the experiment's complete. So even the people that are, you know, maybe measuring, I don't know what you would measure with a headache pill, because generally with tomatoes, you don't have to worry about the tomato plants having bias, but maybe the people that are measuring those tomatoes might have a financial stake in a certain fertilizer. So really, it's going to be better to have the technicians not know which one has the fertilizer A and B and which one is even the control. And it would then be double blind because the tomatoes don't know. But with the headache, it'd be the person handing you out the pill and asking you questions and taking your questionnaire and so forth, as far as a double blind experiment. If you were having a weight loss drug, then the people that would be weighing the subjects, those would not know which drug they had or whether it was drug or placebo. So here's an example of a double-blind experiment. 100 people, subjects, participated in an experiment to test a new headache medication. The subjects were divided into two groups, a control group that was given a placebo, and an experimental group that was given an experimental headache medication. All experimental subjects were told to take one tablet every time they had a headache and to record how they were feeling half an hour later and then a half an hour again after that, after taking the medication. So in half an hour and then in an hour. The experimental subjects didn't know if they were given the medication or the placebo. The technicians giving out the medication and collecting the data didn't know if they were the actual subjects taking the medication or if they were the controlled group taking the placebo. So both the technician and the subjects were blind to the treatment. So it is a double-blind experiment. So the next thing you do in your steps of scientific method, so you've designed and performed your experiment, you're going to record and analyze the data. So the data is the observations and the measurements. And I've heard people pronounce it both data and data. I don't care which one you use. The heights of the tomato plants, the number of leaves produced by each tomato plant, the number of tomatoes produced by each tomato plant were recorded. I would also take a dry weight of the tomato plants, and then I would take the weight of the tomatoes. Data is often analyzed using graphs and charts. So you have two types of data. Quantitative data. Those are observations that use numbers or quantities. So you can remember that quantitative data uses numbers or quantities. And these are measurements or counts. So your heights of the plants, how many centimeters or inches, but in science you usually use metric system. How many centimeters tall are they? How many leaves do they have? How many tomatoes do they have? What is the mass of the tomatoes, the average mass that each plant produced of tomatoes? Qualitative data are observations that do not use numbers. So is a headache alleviated or not? Maybe a color of a solution. Do bubbles form or don't bubbles form? If you want to measure the gas produced by the bubbles, that would be quantitative. But whether or not bubbles form, that's qualitative. Also, I was thinking about these tomato plants. You could also record the color of tomatoes. Maybe some are nice and red or you know, red-orange, and some are like yellow and have spots on them. The color would be qualitative because that's a quality. So you use instruments like rulers, balances, things like that usually for quantitative, and then your data is in numbers. Qualitative, you might use your senses a little more, like, oh, these tomatoes taste great, oh, these are nasty, things like that. The next step of the scientific method is to accept or reject the hypothesis. So the hypothesis is rejected if the results do not support the hypothesis. It's always rejected then. And you know what? If your hypothesis is rejected, then that's it. You have to just go and get a new experiment with a different hypothesis. Scientists can form a new hypothesis, then they can test that experiment. If the results support the hypothesis is accepted, and then the experiment's repeated. Because you want a large sample size and you want the experiment, if it's really going to prove a hypothesis, it needs to support it over and over again. So that's really important. If you ever get a big experiment, maybe if you have one plant and it's just a dud plant, that's one thing. But if you have a big group of plants and they're not supporting your hypothesis, there may be something else causing the difference in growth. So the scientific method, you make an observation. Well, really, before we had you ask a question. Well, you make an observation, then you ask a question, you research it, you form a hypothesis that answers the question. Okay, then you design an experiment and you record and analyze the results, and then you accept or reject the hypothesis. By the way, notice this is a little bit different than the order we have it. You can look, and it depends on the source. Sometimes they're just a little bit different, but basically it's always the same. There just may be a tiny bit of differences. Oh, that guy moved. So the scientific theory is a well-tested and widely accepted hypothesis. So series are supported by extensive evidence. And with a theory, then the hypothesis that it originally was has not been disproven. So the cell theory, all living things are made up of cells, and new cells are formed from pre-existing cells. That's the cell theory. Scientific laws have been tested repeatedly over a very long period of time, and they have never been rejected by experimental data. So the law of gravity, let's see this. Is it going to work? There it is, the law of gravity. But they're all falling down, none are falling up. So now we've looked at scientific method, and we're going to look at the difference between scientific inquiry and scientific method. And there's similarities as well. They're both ways to study science, right? So both scientific method and scientific inquiry are used to answer scientific questions. But the scientific method is a specific series of steps designed to answer one question at a time, okay? So you do this whole experiment to answer that one question or one problem that you have, and you have the hypothesis for the one question or problem, you know, that you think is your educated guess to the answer. And then you follow this series of steps. It's in a line, like a one-way defined order of steps. Communication of the experimental results occurs at the end of the scientific method. You know, that's what they say, and that is overall. That's when the scientist publishes their results and so forth. But a lot of times you do have several scientists collaborating on experiments, and they will communicate throughout. But you don't really get the results till the end. The scientific method is essential for laboratory research and for writing scientific reports. So if you're in a science class where you're doing lab experiments and you're turning in lab reports, it's written in the scientific method usually, okay? If you are trying to publish in a scientific journal and you're doing a study for that, that is going to be following the scientific method. So this right here is the scientific method, okay? And you see, ask a question, do some research, form a hypothesis, design and perform an experiment. And, you know, sometimes when you set up your experiment, it takes a while to get your experiment working. So you may have to redesign it. But then you record your data, you analyze your data, you draw conclusions, and does you reject or accept the hypothesis, and there's your communicate your results. So scientific method. And you know what, you can put these steps in a circle too. You have a beginning and you have an end, but there's the results. But there you go. So now we're going to look at scientific inquiry. Inquire is to ask. Inquiry is a search for the truth, for information, or for knowledge. So anytime you're asking, that's what you're doing. The scientific inquiry includes all of the ways that people study the natural world and seek to answer scientific questions. Not just one way, all of the ways. The scientific method is included in scientific inquiry because it is a way to answer scientific questions. Communication is essential throughout the process of scientific inquiry. Scientific inquiry helps students develop critical thinking abilities, and it enables students to think and to construct knowledge like a scientist as they develop inquiry skills. So a lot of times with classes, when you have older students, you might be using the scientific method a lot more. But with younger students, you may be using scientific inquiry to help the students develop these skills. Inquiry skills include observing, predicting, measuring, comparing, classifying, and making models. So observing is using your 5 senses to acquire information. So you may just have students look at some plants and they're counting some leaves or some tomatoes and so forth. Classifying you might sort things into groups based on similarities. So you may look and say, okay, we're going to put all these animals into this group because they have vertebral columns, so they're vertebrates. And then you switch the warm-blooded and the cold-blooded, okay, the endotherms and the ectotherms. And then here's a group that has hair and mammary glands and produces milk, at least some of them. So the females generally. So there's your mammals. Here's the birds. They have feathers and they're warm-blooded and they have, well, lay eggs. Here's the cold-blooded ones. You might have fish and reptiles and then amphibians. And the fish, by the way, are split too into jawless fish, bony fish, and cartilaginous fish. So there you have that. That's classifying or sorting things into groups based on similarities. There is a lot of that in science and a lot of observing. So for scientific inquiry, you can see there's a lot of questions and there's a lot of things, communicating with others, the observing, defining the problem, and maybe they're all a little bit mixed up and you have your questions in the middle sort of, and you can go back and forth between these different parts of it, of science. Whereas with the scientific method, remember, you started with your question and you went in a straight line. And so here's a table. Here's scientific method. It's linear and has a defined order of steps. You answer a single question. The results are communicated at the end of the experiment. And this is essential for reports, scientific reports, and scientific research. Scientific inquiry, it's nonlinear and it's more fluid. It may end up generating more questions. Now this is in our table, it's on this side, but also that happens a lot with the scientific method. Communication is a key component throughout. And this is essential for scientific communication. Sometimes you have a little bit of both mixed in here and there. Like I mentioned, scientific method is a type of scientific inquiry and so forth, and that you can have communication throughout, but your results really, you publish them for the world at the end of the experiment. So maybe the people with you're working with, you have them more throughout. But this is a good table that I would use for maybe learning this for a test question and in general. And here's again the same picture, and everything's going back and forth to each other with questions in the middle. The scientific method, your questions at the beginning, and your results and your reporter at the end. So we know what science is, the pursuit of knowledge of the natural and physical world through scientific method and scientific inquiry. But we also want to see what technology is. And I know a lot of you are familiar with technology because we are so involved with it every day. So a scientist may study atoms and chemicals, maybe the human body or certain plants, to learn about the plant and so forth. Some scientists discover new species of organisms. They learn about the organisms and they look for relationships between the organisms that they discover and between the organisms they discover and known organisms. So scientists might discover a whole new species of plant that no one ever knew about before. And then the scientist may find that the plant produces a compound that's toxic to some bacteria. So all of that basically is science at this point. Technology is the application or use of the scientific knowledge to create a new product or processes intended to improve our efficiency, our chances for survival, our comfort level, and our quality of life. So science is just learning about something. Technology is using that science to create these products that help us out, okay? So that scientist that discovered the plant in the example on the page before, they may end up producing an antibacterial product that can be used by people to keep surfaces disinfected. Okay, so that because that plant produces something that kills bacteria. they may end up producing a new antibiotic with that. And all of that is technology. So here's some things that you see. One of them, you might have seen DNA electrophoresis. So you can basically look and tell how organisms are related by looking at their DNA and sequences in them. And being able to run that DNA, cut it with enzymes, put it on a gel, that is all technology. Right here, a lot of people might be more familiar with this. Sometimes people with diabetes, they might have this glucose sensor and transmitter, and then they actually can use that and read it on their phone and see what their glucose level is. And then some of them might have an insulin pump, and then the pump can pump in insulin based on how much glucose they have in their bloodstream, or maybe they'll just hit the pump a couple of times depending on how much glucose they have in their bloodstream. But this is definitely technology. My older sister had type 1 diabetes, and she was 6 years old. I'm trying to think when this was, about 1968. And they did not even have the little lancets where you test your blood sugar that they have now. Like now you can check your blood sugar and have a little monitor. They didn't even have that. She just checked her she checked her glucose in her urine every morning and every night by peeing in a test tube and then putting a little pill that turned a certain color if there was urine in it. So that was technology, but not anything like the technology they have today. So here's the differences between science and technology. So science helps explore new knowledge systematically through observation and experiment. Technology is application of scientific knowledge for different purposes. So you use your knowledge to do something. The effect of science is useful for humankind. Technology, the effects of technology can be either useful, helpful, or harmful. Science focuses on discoveries. Technology focuses on inventions. Methods require theory development, analysis, and deduction for scientific methods. Technological methods you're required to review and integrate a design. So with science, the basic principles of science remain the same throughout. Technology changes with every passing day. Every day there's new technology. It's crazy. If you've ever worked in a lab, like I worked at the Jackson lab for just about 4 years because I mostly like to teach, but I thought I would work at the Jackson lab and see if I liked that, and it was okay. But it was amazing just in the 4 years I was there. Then they got a next generation DNA sequencer in the lab that I was at, and just one person there was using it, maybe and some managers, but one of the people I worked with, she got to use it. But that you ended up where you could check an organism's entire genome maybe in a few weeks, and now you probably could in a few days. But when I was in school, there was something called the Human Genome Project, and this was a big collaboration of all or most of the countries in the world trying to get the genome of a human being, and they all worked together for decades on it. So it's amazing now that you can get that done so quick. Now, if you want to see what kind of dog you have, you can just buy one of those little kits, and you can take a swab and send it somewhere, and they'll tell you that you thought you had a Maltipoo, but it's really a Chihuahua Malti Shih Tzu or something like that. Anyway, with a tiny bit of Shepherd, you can get one of those for yourself too, and you can do your Ancestry.com. Actually, my cousin found out she wasn't my cousin. So you find out, and then we found out we had other cousins. So science is used to make predictions. Technology simplifies human life and fulfills needs of people. Goals are achieved through the scientific process. Goals are achieved through key technological processes. Science focuses on understanding of natural phenomena. Technology focuses on the understanding of the built environment. But I mean, you can look now when you see the solar farms and such on the way to the Alphon campus, then definite technology. But science was used to be able to have that technology, or that technology uses science to be developed. So science is acquiring knowledge, whereas technology is applying that knowledge to produce a product. More scientific knowledge leads to more new technology, and more new technology leads to more scientific knowledge, and more technology and more science knowledge and more technology. So they're all sort of wrapped up in each other and leading to each other. Many scientific experiments would not be possible without technology. All of the equipment used by scientists for the experiments are technology. So your microscope. I mean, even if you look at your little dissecting needles that someone made, you know, I mean, those would be technology. And I would think maybe recording something on your lab notebook, but now you can record it all like on an iPad or in a computer. You can make an Excel sheet. So here is science. Here is technology. Science informs. Technology demands more science. Technology makes life easier. Science benefits from technology. Your science seeks to improve society, and society demands more science. I mean, it's all wrapped up. But technology is a user application of scientific knowledge for a specific goal or purpose. Our next topic is scientific information versus pseudoscientific information. So the word pseudo means false, okay? So this is false scientific information versus science, okay? Sometimes it's really hard to tell what is science and what is pseudoscience, or what is science not at all. So people will slant information and make it seem like scientific evidence supports the hypothesis because they want your money. I mean, that's usually what it is. They want your money. They want to sell you something. Or maybe they want you to vote their way. Either way, they're going to slant the information that makes it seem like scientific evidence supports a certain hypothesis because of whatever reason they have. So you need to make sure that you know the difference between what is science and what is fake science. So pseudoscience, that's theories, ideas, or explanations that are presented as scientific, but they're not actually derived from science or the scientific method. So astrology, you know, when you look at the stars and you're like, okay, the stars are lined up and this is, I'm a Virgo, you know. Astrology is considered a pseudoscience because a connection between the positions of the stars and human behavior hasn't been proven scientifically. Notice I said it hasn't been proven scientifically. I haven't said it won't be proven scientifically, but it hasn't yet. So it's not science. That is false science. Another thing that's important is to know whether your sources are real sources from real science or just some fake source with someone trying to sell you something. So, and before you answer the discussion, I hope you read this part of the notes. But primary sources, this is the original research written by the scientists that performed the experiments and then published, usually in a scientific journal. So you'll see like Nature, Science, American Journal of Physics, JAMA, things like that. Primary sources have been peer reviewed. Sometimes they might publish them a little early and then they'll say not peer reviewed, but then maybe a month or two later they'll come out peer reviewed. So journals that publish primary sources of scientific research include Science, Nature, the Journal of American Medical Association, it's JAMA, it's not about jelly, it's American Medical Association, the Journal of Molecular Diagnostics, and Human Molecular Genetics, all of those and many more are primary sources. So here is a primary source. PubMed is a really good website for finding them. And you know, when I was in college, well, you didn't have websites yet. Okay, really, you didn't have websites. I don't even think there was email, honestly, when I was a freshman. But there were computers. Just I only knew one person that owned his own personal computer, and that's just because he worked at Best Buy. But now, before you would go into the library and they had the journals that they would buy and you could look at them. But now you go online, and I'll tell you what, if you're doing any research, some of these that'll say that you have to pay like $50 to see an article. Don't do that, because if you're a student at KVCC, then they can get access to a lot of these articles for you. So contact the library if you can't figure out how to get them yourself. But here's Human Molecular Genetics in PubMed, by the way, it's really good. Some of them you'll see and it'll say that you can click on something and download the free copy. But this is characterization and behavioral and neuromuscular junction phenotypes in a novelic allelic series of SMA mouse models. And you can see all of these are the authors that were all working at it. They were all at different places. Usually the names are in alphabetical order by last name, but sometimes, like this person, she was a primary author. So she did all of the writing, and people from, I know, Jackson Lab in California and a lot of other places, you know, all gave her, you know, the information that they got and that she put together, and she wrote the paper. So primary author here. And this is the person who's the PI, the primary investigator, right here for most of it. So, but otherwise they're mostly alphabetical by author, last name first. Most primary sources of scientific study will have the following sections in this order: title, authors, then an abstract. And an abstract is just a little bitty summary of the article. That way a scientist can just read a couple of paragraphs and it'll tell you a little bit about the article, and then you'll decide whether it's going to be worth your time to read the rest of the 20 pages of the entire experiment and interpret that. So, because sometimes they're really hard to read, some of these. But so there's the abstract first, which is a summary. Then there's the introduction, which is background material, the materials and method. This is your step-by-step experimental procedure, okay? Then your results section, where you list all your results, and then you'll analyze them, and you'll have that in your discussion, and then have your conclusion and a reference section. Notice this is the same order as the scientific method or lab reports for a class. It's all in the same order though. You have your question, you do your background research or hypothesis, you test your hypothesis with an experiment, and you analyze the data, and then you determine your conclusion. So there it all is. So when you are looking at something, then you look at a primary source, it should look like the scientific method, okay? And it should be in the order of those steps. So your title, usually it's not a cute title. When you have a scientific paper and a primary source, it's not going to be a cute title. It's going to be a title that describes the experiment that was performed. So ordered in deterministic cancer genome evolution after P53 loss. Okay, so that's like a certain gene you're talking about here that you have a lot of issues with it with cancer. Your control of working memory in rhesus monkeys, and then here macaca mulata is the I'm not sure exactly how to pronounce that, but that's the rhesus monkey scientific name, which, by the way, you'll notice the genus name is capitalized, the species name is lowercase, and both words are italicized. So if you turn in a paper with a scientific name that's not written this way, you'll lose at least a point. So right here, elotuzumab as a novel anti-myeloma immunotherapy. Okay, so here's one published online 2017, and this in human vaccine immunotherapy, maybe human vaccination immunotherapy, but there's a journal all on immunotherapy in humans. So this one is in Nature. But see, that's what your title does though. It explains the experiment or describes the one that was performed. Then the authors I mentioned before listed in alphabetical order by last name or with primary investigators and scientists with most responsibility listed first. Although here she was the primary author, she mostly wrote it and she did a whole lot. And then here's a primary investigator right here. And look, here I am. So I just happened to be at the right place at the right time, and I did a little bit of the research that was in there. Actually, I don't even think it was research. I don't remember exactly, but I was at the right place and at the right time, that's for sure. And here's all the people I worked with here. And look, they're all together in the same lab. So all of us were in the same lab. This person here, she raised the mice, so she was in the mouse room. This person, I don't know exactly what she did, but she sat right next to me. So here is the abstract. This is your summary of your experiment. Okay, this is a different experiment. This was in the optimization and characterization of a bone culture model to study prostate cancer or bone metastasis. So how prostate cancer cells can go and cause cancer in bone. And then here's the abstract that just gives you a summary. Now here's your introduction. This is the background material and the hypothesis. So anytime you write a lab report, you'll have that. And I have tried to write some of your lab assignments in the same order. So where you have the introduction. I haven't put abstracts on them all though, because they're not for publication. But here it contains a background, and they're not long enough either, but contains background information and the hypothesis. Then your material and methods section, that includes the materials needed to perform the experiment and the steps of the experiment that were performed in the study. So sometimes you'll have a list of materials and then you'll have the steps. Other times you'll have just all the steps with the equipment listed throughout it. And then sometimes they will refer to someone else's experiment, and I don't like that. They'll have a site. Then you have to go look back at that person's experiment if you want to do it. And then theirs may cite another, and then you have to go find the other, and then they may cite another. So I prefer that you just list your own steps. So another scientist should be able to repeat the experiment using these instructions. There's always some issues with the equipment differences being a little bit different, but still. The results section. This contains the data from the experiment. So here's your tables, graphs, your pictures, and your charts might be in the results section. And then in the discussion section, the data is going to be analyzed and discussed. So your results section just has the results. The discussion section, that's where you analyze it and talk about it. And if you have unexpected results, then you try to explain those. So sometimes you may realize later that you accidentally put the wrong amount of sugar in your sugar water solution. So you might mention that in your results, or in your discussion, excuse me. Then your conclusion is a summary of the experiment. This is what was learned and what was important. And also, you need to say whether it supported your hypothesis or not. I need to add that in the notes because that's real important. You need to say whether this whether the data supported your hypothesis. Then your reference section, this is a list of outside sources used for the experiment. And you might need outside sources to explain your background material or to develop your experiment. So you need to make sure to give them credit. And usually it's in APA format lately with science. Now, those were all primary sources, so there's a lot of information on primary sources. Secondary sources are works that analyze or interpret your primary sources. So examples of secondary sources include review articles and critiques of your primary sources. Secondary sources, a lot of times they're easier to read. They might have a broader overview and maybe more background material, but they're going to have less technical methodology, exactly how something was done. Secondary sources were created by someone that did not experience the research firsthand. And you'll see sometimes it'll have review, like right here it's written the Lancet review. And that way everyone knows it's a secondary source. Now your primary sources are considered the most valid because there should not be bias in them. Not nearly as much. You can look and you can see the exact method and procedure that the researcher used, and then you can see the exact results. And then you can read the discussion, but you can look at the results and you can interpret yourself and you can tell. But with a secondary source, you don't necessarily see all that. You see an overview of it. So even though secondary sources are good in a lot of ways, they're not as valid as a primary source. Now, tertiary sources are summarized or condensed versions of primary and secondary sources. So someone might go and say, here's a secondary source, a whole bunch of those. So we're going to go and write a textbook using a bunch of secondary sources. Now, the thing is, a tertiary source is a good place to get a general overview of a subject. Okay, I mean, you're not going to go to take, you know, a biology class or an anatomy and physiology class and look at, you would have to look at so many primary sources. So you would have a textbook or a dictionary, encyclopedia, handbooks, things like that. So they're good for being able to get a lot of summarized and condensed versions, but they're still not a primary source. So you have to keep that in mind. And you can double check the validity of your tertiary source by going back and looking at a primary source. So here they are: primary literature, original research. They're peer-reviewed. They're in journals. They could be dissertations, technical results, conference proceedings, general experiments. Here's your secondary literature. So generally reviews of primary sources. And then your tertiary sources, summarized in condensed versions. And you can use references to both primary and secondary resources. And it's a good place to get a general overview, look up some facts. But you have your textbooks, dictionaries, encyclopedias, and handbooks. And it's interesting because this is why you see a lot of variability in different textbooks, like the steps of the scientific method. Well, you know, some people just do those a little bit different. Some people say that the hypodermis is the third layer of skin. Other sources say that it's not a layer of skin at all, you know, that it's a layer below the skin. So, you know, here's other sources. Other sources. I should turn on the camera, then you can see I'm doing air quotes. I don't know why the camera goes away, but that's okay. But your other sources. These are news articles about primary sources that are on the internet, TV, radio, newspaper, or magazines. Now, sources other than primary sources may make the scientific information appear to mean something that it doesn't mean. So that's pretty important. Okay, like this right here. Snake with human head is found in Arkansas. I mean, you know, is this real? I doubt it. You might want to look at Journal and Nature or something like that, and maybe you'd find it in here. Here is Dr. Quack's tapeworms. Like they spelled it, you know, but it does sound like quack. Get rid of life-threatening fat without surgery, exercise, or dieting. And they actually really used to sell these tapeworms. They were like little pills that had a tapeworm in it, and people would take it. And of course, they make you lose weight. I mean, you have tapeworms with suckers and hooks in your intestines, eating all your nutrients before you can absorb it. But here is this head transplants, and it can be done on humans now. And there's Elvis. Okay, head transplants. I'm not going to be looking at the Inquirer to see if they can do head transplants now, because according to the Inquirer, I'm sure they can do anything they want to say. So when you look at scientific scientific sources, you need to be cautious of the following. Anecdotal evidence. This is evidence given by one or two people, okay? Always look for studies with a large sample size. So you may see someone and they're on TV and they're like, oh, my wrinkles all have gone away since I used this, you know, this skin cream with Botox in it or something like that. I don't know. Whatever it is, but it's one person. This has worked miracles for me. I look 10 years younger. You know what? Maybe it did work for that one person, okay? But that doesn't mean it works for another person. That's just one person, this anti-aging data here, okay? So a hypothesis should always be experimented with a large sample size, not 1 or 2 or even 10 people. We're talking hundreds to thousands of people, because there's a lot of people. And really, 100 people is not that many people, because they can pick the 100 people that they want, that maybe it's going to work for those. So when looking at scientific sources, be cautious of sources that have something to gain. So is the source an ad or an infomercial? And now it drives me nuts. You'll watch this thing that says, oh, this ice trick that none of the doctors want you to know about losing weight. And then you'll have to watch an hour video, and then they try to sell you these pills, which before they were just telling you it was an ice trick. I bet there's people besides me who've clicked on that. But there you go. Are they an ad? You don't think they are at first, but then they end up being one. A source may be a medical source, so that might be okay if it's like a doctor's office. It may be an advertisement, and you know they're trying to sell you something. It might be both. It might be a doctor's office with an advertisement. So, does the media source running the story on a new drug or product have some type of behind-the-scenes connection to that product? And that's kind of hard to find out these days, but that's something that really could affect an article that you see on something. Also, does the source itself have some type of agenda? So make sure that when you're seeing any type of science story, then you consider all of this and be cautious of it. Also look at out-of-date sources, okay? Because maybe there's something that was true 20 years ago, you know, the information was valid. Maybe this medication was the best one out there 20 years ago, but not today. There might be something 10 times better. Maybe they found out that something else was causing the wrinkles, not just, you know, some certain water you're drinking. Maybe it's the UV rays of the sun. Who knows? But you need to look at some outdated, look at the dates on the sources. That's why when I have you write a paper, you do a literature review paper this semester, and your sources need to be within 5 years. Also, does the reporter or the source understand what they are reporting? And I'll tell you what, I have seen all kinds of people reporting on a source, a science source, make mistakes left and right. And it's really sad because no one knows they have an error, that they don't understand what they're talking about. But if you're a scientist, you can tell that they have no idea what they're talking about. So that's really important too. Maybe the information might be valid, the original information from the primary source, but the person reporting it really doesn't know what they're talking about. So it doesn't come out sounding like it really is supposed to be. So also look, is the scientific discovery itself controversial? And look, flat and round. And now you might be a flat earther, and that's okay if you are. And then there's round. But you know, before there was some guy saying that the earth was round, and everyone was like, no, no, you've got to be wrong. Everyone knows the earth is flat. And that was very controversial. And now it's like everyone thinks it's round except for the flat earthers. But anyway, are there many conflicting and confusing stories? Well, that could be a problem. So if you have many conflicting and confusing stories, you really need to look at the primary experiments and see what they say. Are the scientific papers even conflicting and confusing? And if the scientific papers are conflicting and confusing, you really need to look at the experimental methods that they use and how they're recording the data. Because remember, double-blind studies are really important, and how people record the data and how they analyze the data is really important too. So make sure you know what information is true with science, especially when you're making decisions that affect your health, okay? And now it seems like they expect you to understand a lot more, or to be a lot more involved in your health decisions, and you need to make sure that you understand those decisions. Look at the primary source or primary sources, look at all aspects of the data, and examine the fine print. If you're not sure about a source, look at the references. There should be more than one reference usually. But if a source has real experimental data supporting its claims, then the primary sources of that should be listed, and then you can look those up. And I've noticed that when I go on to sometimes like WebMD or something like that, and I'll read something, and then I can look at the bottom and find the sources. If you can't see the sources at the bottom of that article, then there is a problem. And maybe it's valid, maybe it's not, but why aren't they showing you the sources? I don't know. Read the fine print. Is a new product FDA approved? And that's really important. Is it FDA approved? And if it is FDA approved, was there something to gain? Like there's some question about aspartame, and there always was. Like back in the 80s, when aspartame was approved, and then all the soft drinks had aspartame, saccharin was switched over to aspartame, and then there's all this. Even now, people that are thinking, well, aspartame is a lot more dangerous than saccharin, which is what was in them before. So who knows, but that's definitely, you need to look at that. Many herbal supplements do not have to follow the same guidelines as medications. So you don't have the FDA approval because they're not a medication, they're a supplement. So that's something to be aware of. And I'm not saying that herbal supplements are going to be a problem. Or is bad because of that. I'm just saying they don't have the same guidelines, so you need to pay attention. I think some of them are great. Make sure you look at the reviews, and reviews that aren't from the source. That's really important, because nowadays, when I go to, like before, you would go and look at reviews and you'd see all these people with different reviews and different opinions. Sort of like, at least on Amazon, you have a lot of different opinions. But a lot of times when I go Google something and ask for reviews, then I'll end up getting something that I find out is from the actual source. So it's like they end up getting Google Ads or something and getting the first bunch of reviews that they're going to have. They're written literally by the source. It drives me nuts, because I'm like, well, that doesn't really tell me anything. So that is the end of our unit on scientific inquiry, science, and the scientific method. If you have any questions, feel free to email me. Good night.